Thank you very much, and thank you for the invitation. I'm really excited to get the opportunity to present to you some of the results of our studies we've been doing on sort of the overlap between psychiatric disorders and aging using different methods, both neuroimaging and proteomics. I have nothing to disclose, and sort of as a next step to the presentation you just heard, I would like to show you some studies that are looking at the intersection of serious mental illness and aging, and I'll quickly introduce the concept to you. You'll see some overlap, but also some additions to the neuroinflammation hypothesis. And then I will show you some of our work we've been doing on the imaging side, where we're looking at brain structures. Then I'm going to show you some of our work we have been doing on like the peripheral side using proteomics, and then some preliminary data on how to bring that both together. And in the end, I'll show you next steps and limitations, and I'm also looking forward to questions. So aging, of course, isn't a disorder or disease-specific process. However, on the one hand, with an aging population, we are going to have more individuals affected by psychiatric disorders. And on the other hand, there is evidence that individuals with psychiatric disorders are more vulnerable to aging, or at least a subset of individuals present with premature aging, with accelerated aging. And epidemiological studies show that psychiatric disorders are associated with adverse long-term outcomes, including clinical deterioration, cognitive deterioration, and also increased risk for dementia. And in addition, and we've heard about this in the keynote earlier, we also see an increased risk for medical comorbidities and then actually increased mortality due to general medical conditions in psychiatric disorders. So from that evidence, it's quite clear that psychiatric disorders and this like increased vulnerability to aging affects both the brain and the body. However, traditionally, we're not really looking at this interaction. We're looking mainly in psychiatry at the brain. And we have many measures, most of them indirect, to look at the brain, including imaging, which is what I'm going to focus on today. On the other hand, in general medicine, we have peripheral markers and lots of successful studies of that. However, we have not really translated these findings into psychiatry. And what we're sort of missing is looking at this from a psychiatric lens. And I'm going to show you some of our efforts where we're trying to do that. So let's start with the brain, and first the white matter structure. And the first study is the same study Marek introduced earlier. He showed you the free water findings from this study. What we wanted to do there was to look specifically at white matter in schizophrenia. And we were interested in looking at white matter pathologies across a wide age range. Ideally, to do that, you would use longitudinal data. However, at this time point and today, there wasn't any big longitudinal study available. And there was also no cross-sectional study available which was large enough to look at these like age trajectories. So as Marek said, in a first step, we had to combine data from many different sites. Altogether, we had 13 sites and over 1,000 individuals. And this is quite difficult when doing imaging, especially diffusion imaging, which we use for white matter, because it's highly dependent on the scanner. So my colleagues in a first step developed an algorithm which allowed us to combine data. This is freely available, and if you are interested, please feel free to reach out. In the next step, we extracted different measurements from this harmonized data set. And Marek showed to you the free water, which is looking at extracellular pathologies. And I would like to focus on FA, which he briefly introduced, which is more closely aligned to cellular pathologies. A little simplified, we often say that higher FA means better organized white matter or higher white matter integrity. So we modeled FA across our age range of 14 to 65 years, and what we found across the board is that individuals with schizophrenia presented with lower FA. So we saw evidence for lower white matter organization. When we looked at these cross-sectional trajectories, we found a mixture of changes indicating more developmental processes, changes indicating maturational processes, but also evidence for degeneration or accelerated aging. And supportive of that idea, we then in a follow-up study looked at associations with clinical measurements, and we found very few, but one of them was that a longer duration of illness was indeed associated with more white matter abnormalities. And what you can see here on is that we then, in a cross-sectional model, also showed that these white matter changes were at least partially explaining the association between a diagnosis of schizophrenia and cognitive deficits. So taken together, these findings support the notion that white matter is indeed very relevant for schizophrenia, and they also provide some evidence for this idea of like accelerated or premature aging in white matter and the relevance for cognition of that. Another way of looking at this idea of premature aging in a cross-sectional data set is the so-called brain age approach. Briefly, the idea here is that you use a large data set of healthy individuals and you derive some measurements from that data set. Many models use gray matter, so gray matter volume or gray matter thickness, and then you use these measurements to build a model that predicts chronological age in your data set. And then you can take this model and apply it to different data sets, including data sets with individuals with psychiatric disorders. And then the model gives you a predicted age in your new data set. And the difference between the real age of a person and their predicted age is what we call brain age or brain age gap. And again, a little simplified, we often say that in adults, it is better if your predicted age is younger than your real age. So you have a younger looking brain. And it's worse if your predicted age is older than your brain, so you have than your real age, so you have an older looking brain. This method still needs to be validated in longitudinal data sets, but there are studies out there which have shown an association with lifestyle factors, including, for example, alcohol, smoking, or pain, which are all associated with an older looking brain. Or on the other hand, education, physical activity, or playing music, which have been all associated with a younger looking brain. And while most evidence is in classical neurodegenerative disorders, there's also some findings in psychiatric disorders, including, for example, depression or schizophrenia, which suggests that individuals with psychiatric disorders indeed present with older-looking brains. Taken together, I hope I showed you some evidence of this idea that individuals with psychiatric disorders are more vulnerable to aging, and you can see that in the brain. And with that, I sort of would like to switch to the periphery. And you probably know there's like many studies out there which are looking at single proteins in psychiatric disorders. As I said earlier, these studies have not really been translated into clinical care. And I believe that one reason for that is that we are often looking at single proteins or a single system. However, if you're taking a measurement peripherally from the blood, it's quite apparent that this is going to be a messy measurement. It's not going to be very specific for one psychiatric process. I don't think that's really a disadvantage. It just means that we need to look at these measurements differently. And one approach which is quite popular in general medicine and actually in the aging field is that of looking at cellular senescence. And cellular senescence means that cells stop replicating. This happens throughout the lifespan. It's normal, it's essential for normal development. However, there is evidence that both as a reaction to stress and then also with aging, more and more cells turn into these senescent cells. And what's interesting about this is that they don't only stop replicating, they also show body-wide effects through the expression of a set of proteins, the so-called senescence-associated secretory phenotype proteins, or SAS proteins. And these proteins include proteins related to inflammation, but also to cell communication, to tissue remodeling. They have been associated with many medical conditions that are prevalent in aging, and indeed it has been suggested that a difference in senescence might explain why some individuals are overall more vulnerable to these disorders associated with aging than others. And why it's interesting for us, and if we think about this brain periphery again, is that there is evidence that brain cells show a very similar phenotype. And as you've heard earlier, there's also evidence that the blood-brain barrier permeability increases both in psychiatry and with aging. What is the problem of this concept? Why has it not been used in psychiatry that much? The problem is that a lot of the studies to date are preclinical studies, and it's a quite broad concept, which also means we don't have one specific set of markers, and it is not as clear what markers should be included and what shouldn't be. Besides the sort of preclinical work, there is some work in depression where researchers are trying to characterize which sets of protein might be a good approximation for this senescence process. And the studies, while it's only a few, have actually quite consistently shown an increase of these SAS proteins in depression, both in younger individuals and in older individuals. And we were interested in following up on this, and we're very lucky to sort of get access to the baseline data of a clinical trial. This is a clinical trial in late life depression. You can see the age range is between 60 and 90. And the advantage of this baseline data is that it was very, very well characterized. We used a similar approach to previous studies and determined a SASP index based on 22 proteins altogether. We then looked at different clinical data, and what we wanted to know is which data really explains this increase of senescent proteins in depression. And the categories we looked at were categories related to psychopathology, so depression and anxiety variables, then variables related to cognition, and then variables related to physical health. And we grouped all of them using factor analysis and then looked at the association with demographic variables and the SASP index. And this summarizes our findings, and sort of as a sanity check, yes, if you're older, you have more SASP proteins. Also, if you're male, you have more SAS proteins, and we see a weak association with cognition. We don't see an association really with these psychopathology variables, but we see that in individuals with depression, this increased SAS proteins is driven by their physical health. And as I said, I think this aligns very much with this idea of depression is actually a disorder that doesn't only affect the brain, but also the body, and we have to look at this interaction if we want to treat individuals and prevent some of these comorbidities. In a next step, we wanted to look at the same proteins in psychosis, and all data I'm showing from now on is preliminary, it's not published yet. This is a very different data set, and we also choose a different approach. So for looking at this in psychosis, we looked at the Human Connectome project for early psychosis, the HCP-EP data set. And these individuals are much younger, the mean age is around 22, and it's a transdiagnostic data set. So individuals were diagnosed either with affective psychosis or non-affective psychosis, with the most common diagnosis being schizophrenia, schizoaffective disorder, bipolar disorder. We also decided to not only focus on these 22 proteins, but to look at more proteins. So we altogether had over 300 proteins which have been associated with cardiometabolic health, cardiovascular health, and inflammation. And as you can see here, the sample size is much smaller than the one I've previously showed, so these results really are preliminary. In our first step, we looked at the SAS proteins, so we looked at the pre-selected proteins. On the left, you can see the same proteins as I've just introduced for depression. And in the meanwhile, there's been some literature coming from preclinical studies where there's an effort to characterize the SAS proteins as well, which is called the SASP Atlas. So we also included that. And there was some overlap between these two, but there's also some differences. And what we found is we found an increase in individuals with psychosis, even that young, so they are 22. And then, which you can see on the slide, we also see that the single proteins more strongly correlate with each other in psychosis compared to healthy individuals. And this supports this idea that this really is a construct, so that it does make sense to look at them together rather than looking at single proteins. As I said, in the next step, we wanted to sort of leverage the whole proteins, not like looking as based on the literature, but taking a data-driven approach. And we ran group comparisons, and we indeed found an increase of some proteins in individuals with psychosis. These include TNF-RSF9 or renin or leptin, so both inflammation and more cardiometabolic health-related proteins. But again, this is not, like the plot I'm showing you is not that useful. It's not that easy to implement. So in the next step, we wanted to see if there's whole pathways which are changed. And for that, we employed gene set enrichment analysis. And what we found in this sample is that by far the most significant differences were in the tumor necrosis factor pathway. This is an inflammatory pathway again. It's one which isn't as commonly looked at as other markers, and which you can also see several other pathways were significantly different between groups. The ones in dark blue are all inflammatory pathways, and the other ones are more cell communication pathways. In some preliminary analysis, we then wanted to bring that together, sort of the proteomic findings with the brain findings. And this is using the same data set I just introduced, the HCP-EP data set. However, this is only looking at individuals with psychosis. So it's following up on this idea that maybe we have an inflammatory and aging subtype which might be more informative than diagnosis in this sample. So what we did is we grouped our individuals using a data-driven approach based on our protein markers. And we, in that instance, used the SASP Atlas just because this approach works better if you don't have too many proteins. And what we found is we found two stable groups where we find one with a higher expression and one with a lower expression. However, this differentiation is mainly driven by a few proteins. And interestingly, and aligning with the previous talk, all of these proteins are associated with the extracellular matrix dysregulation. And then we also found that in the individuals with a higher expression of these extracellular matrix proteins, we see an increase of free water. And if you remember the previous talk, this is an indicator of extracellular pathologies, which sort of has been associated with inflammation. So this again supports this idea that there is this inflammatory subtype in this instance in psychosis. So bringing these studies together, I hope I convinced you that there is evidence for premature aging in psychiatric disorders, that it's important to look at that. And we see evidence both in the brain and in the periphery. However, we need larger studies to look at the interaction of both. And I introduced to you the concept of senescence as sort of one way of looking at this. And the reason why I think this is a very interesting and promising concept is that there is actually a lot of development for therapeutics coming out of the aging world. And there are several preclinical and by now clinical trials which are looking at senolytics and seeing if we can modify this phenotype. And this would mean that if replicated, and we indeed find that there is this subset of individuals with psychiatric disorders, which is also presents with increased senescence, that we could potentially also offer them some treatment. And with that, I would like to thank everyone who contributed both their data and their knowledge to these studies. You can see this is really a team effort. A special thanks goes to everyone highlighted in blue. And I would also like to acknowledge all our funding sources, especially my funding sources of my 2 mentored awards and the great mentor and advisory team I'm having, with special thanks to Merrick. The most important thank you is to all the individuals who contributed their data to the studies, and then to you. Thank you for your time and attention, and I'm looking forward to questions.